Hi guys, today we will be discussing about the practical of phytochemical screening. Most often, especially from the plant source, we have to uh, screen screen the substances from the plant sources on the basis of the phytochemical present in the extract. So we will be proceeding with today's lecture. Let's start with phytochemical screening of plants' chemical constituents. It's a chemical analysis process. So first of all, we can do several constituents about carbohydrate, glycosides, tannins, saponins. So first of all, we will be separating this plant-based sample into three parts. The sample should be homogenized. The first part that will be taken into a beaker, and second part that will be taken into test tube, and third part into the flask. So first of all, we will be doing the aqueous extraction. So we are adding the water, about 70 ml water in this one, and mix it well. Then we will be heating on a magnetic stirrer with heating facility. In while the sample is going to heat, we are preparing the filtration assembly. So after the boiling process, we are going for a filtration process. We are just applying the pressure on the cotton that we have put into the filtering media. So all of the water aqueous extract that will be obtained. So first of all, we are performing the Molish test for the carbohydrates. So take 2 ml from aqueous extract in a test tube and add few drop of the Molish reagent in it, and add few drops of the H2SO4 through the wall of the test tube. Then rinse, then shake it. If the red ring is appear, it means the sample is positive for carbohydrates. Then for tannins, again take 2 ml of aqueous extract in the test tube and few drops of the FeCl3. Blue color mean hydrolyzable tannins are present, and green color mean condensed tannins are present. For saponins, froth test is used. 2 ml of aqueous extract is taken into the test tube. Shake it. If 1 to 2 centimeter froth is appear, that persists for 5 minutes, showing the presence of saponin. Acid extraction. For acid extraction, take 2 ml HCl in the test tube, cover it for 20 minutes and shake well. After 20 minutes, filtration will be, we have to do the filtration. So shaking and dissolving it, keeping it for 20 minutes. Then filter it. It will prepare the acid extract. We will be performing flavonoid test on the acid extract. 2 ml of acid extract in first test tube. Take it. Same is the case with the 2 ml of acid extract in the second test tube. And one test tube should be for the blank with 2 ml with distilled water. To the second test tube, add 2 ml of NaOH This is the NaOH. And now we will be going to compare the color of both the test tubes. The darker yellow color show a positive test for the flavonoids. Now we will be preparing the alcoholic extract from the sample that was taken into the flask. So to that flask we are adding the alcohol, about 80 ml alcohol we have added into it. Then we are going to cover it, and we will take a rest time of 30 minutes for completion of the extraction process. So after extraction, we will going to filter the extract using the cotton filter again. Then this alcoholic extract is shifted into the porcelain dish, and that will be heated until all the liquid that evaporates. So after evaporation process, that we are getting this dried material into it. To this dried material we are adding 6 milliliter of chloroform and dissolve all the dried matter into that chloroform. And we will be now performing different tests of sterol, triterpenes, anthraquinones, and the anthraquinone glycosides. So first of all, for the alkali test with capillary tube, we are taking the sample in the form of the drop onto the filter paper, and another sample for standard that contain the alkaloids in it. Then we are going to spray the filter paper with dragon drop spray. So the downward side sample was the positive sample. Sterols and turpentines, add 2 ml of alcoholic extract plus 2 drops of acetic anhydride plus concentrated H2SO4 that is added on the walls of the test tube. Through the walls, a reddish color ring means a positive test. You can see here in this test tube. Then sterol and triterpenes. We will take 2 ml of alcoholic extract plus few drops of H2SO4. A reddish brown ring that will show a positive test, as you can see in this test tube. Then anthraquinone glycoside test. We 2 ml alcoholic extract and 1 ml ammonia, shake it well. The presence of the rose red color that will show a positive test. You can see here the rose red color on the top that will show a positive test. Thank you very much.